Nucleic acid purification and amplification. Have you ever asked yourself how do scientists learn the structure of such a tiny molecule as DNA? Do you know how the medical examiners determine the criminal using just a blood sample? How can 20 minutes test tell that you're HIV positive or have coronavirus? How drugs are created? This is all done through the analysis of DNA. There are three main ways to isolate DNA, which we will now look at. The first one is organic acid, the second one is spin column, and the last one is PCR.
Organic DNA extraction method. The first step of the method is cell lysis. Reagents which are involved in the process: salt and buffer. Cell lysis is the process when cell membrane is broken down and DNA is released. Also, EDTA is one of the components of the process. The main role of EDTA is to block DNA's activity. It is an enzyme which lyses the DNA. The next step is centrifugation. During that process, cell debris is usually deleted. The next step is called cell debris denaturation. The protein denaturation occurs under enzyme which is called protease, with phenol or a mixture of phenol and chloroform. Here we can see that RNA can be degraded with the enzyme which is called RNase. The last step is called DNA precipitation. It is the last step in the whole process. Here is DNA is precipitated with the help of ethanol. The aim of the method is to isolate and purify DNA that is taken from blood, saliva, epithelial cells, and hair. Advantages of the process: firstly, the method is very easy, and secondly, it is very cheap. But the disadvantage is that it takes numerous steps and here it is no automatization.
Spin column DNA purification is one of the most effective and fast methods of DNA purification, also called solid-phase DNA purification. First of all, we need to destroy the cell membrane of the sample with a lysine buffer, thereby releasing the nucleic acids. Then, the binding solution is transferred to a spin column and the column is put in centrifuge. The centrifuge forces the binding solution through a silica gel membrane that is inside the spin column. Then we need to wash to obtain pure nucleic acids. The final step is to add an elution buffer to the spin column for washing the nucleic acid. Plant and animal tissues can be used as a sample in this method. The advantages of these methods are that the column-based technology is fairly easy to implement and provides high yield, concentration, and purity of DNA. The disadvantages of these methods include the possibility of clouding the pores with viscous impurities such as lipids and sample residues.
Polymerase chain reaction. Have you ever wondered who invented PCR? It was made not long ago by this smiling gentleman, an American biochemist, whose name is Kary Mullis. He was awarded Nobel Prize for PCR invention in chemistry in 1993. So, what do we need for PCR? First of all, we need this machine that is called thermal cycler. It is quite costly. Then, of course, we need our DNA, some primers to begin the reaction, some nucleotides to build the DNA, Taq polymerase enzyme, mix buffer, and of course PCR tube. Look on the one of PCR cycles. We have our two-stranded DNA and the first step that is heating to 95 degrees. We have denaturation and two single-stranded DNAs. Then we cool them to 55 degrees. That is called annealing. Primers are binding to the complementary triplets. Then the third stage, heating again to 72 degrees. This is called extension. New nucleotides are connecting to the strands. There are plenty of fields where you can apply PCR. I will list some of them. First is mutation screening. Second is drug design. Third is, for example, molecular ecology. Advantages of PCR are high accuracy, comparatively high velocity, and about disadvantages, the expensiveness. To conclude, we can say that methods in the future will lower the cost, become faster, will become more accurate, and will apply more machines. Thanks for watching.